everyone, welcome back to my channel. So we are starting with the chapter Structure of Chromosome, Cell Cycle and Cell Division, and this is lecture 1. And in this lecture we are going to cover the topics like what are chromosomes, discovery of chromosomes, and the last topic, structure of chromosomes. Okay, don't worry about the notes. I have already uploaded the notes of this topic in the short section. Okay, and at the end of this lecture I will be talking about a book which will definitely help you in your academics and achieving your goals. So stay tuned. Let's begin with our first topic, what are chromosomes. Before understanding chromosomes, let's see how chromosomes actually look. Okay, see, this is a normal cell, normal living cell. Okay, why we are taking living cells? Because living cells have different organelles which are present inside the cell. Okay, now this living cell is unstained. I will explain. See, what is a stain? A stain is a chemical, okay, which when we put over this cell, it gives specific colors to different organelles, okay, and because of that stain, we are able to see those structures, okay. If you will just normally observe it without any staining, you won't be able to identify the different structure. But if you are staining, you will be easily able to identify different structures. So that's why before we are using unstained condition, okay, and we are observing under a microscope. And when you observe this under a microscope, this is the cell and this is the nucleus. You won't be able to see anything inside the nucleus because this cell is unstained. Okay, and because of this unstaining, you are not able to see anything. So, nucleus without any structure visible. Okay, just now let's see the other condition. This is the cell, again the same cell, normal living cell. Okay, and this time we are using stain. Stain is a form of a dye. Okay, so we are using stain. Okay, you have seen people using dye to color their hair. So these dye give colors only. Similarly, stain gives colors to different part of the organelles. Okay, for clear visualization. Okay, now observe under a microscope. What you will see? This is the cell. This is the nucleus. Nucleus is always a double membrane structure. Okay, inside the nucleus you will see thread-like components present. These thread-like components are called chromatin fibers. You will see nucleolus, a small rounded body, which is also present inside the nucleus. Okay, and you will also observe these centrioles. The function of centrioles is to help in cell division. Okay, these are the structures which are present inside the nucleus, chromatin fibers and nucleolus. Okay, and it is one of the stages of the cell. So this stage is known as interphase. It is known as resting phase because the cell is preparing for division. We will be studying about all the different stages of a cell cycle, okay, in this chapter only. Now, here I am giving you a hint. See, cell division, it is divided into two phases, mitosis and meiosis. This mitosis is further divided into four substages: prophase, metaphase, anaphase and telophase. Okay, for meiosis you just need to remember that it is divided into meiosis 1 and meiosis 2. The sub stages which are there that are not within the scope of your syllabus. You just need to remember that it is divided into meiosis 1 and meiosis 2. Okay, so the way we observe the prophase stage under the microscope, similarly you may observe any of these stages under a microscope. The cell may be at any of these stages. Okay, now this was just a small hint. Again I am drawing the interphase stage. So this was our interphase stage in which we observe the nucleus, and inside the nucleus we observe the chromatin fibers, nucleolus and centrosomes. This was chromatin fiber. This is nucleolus. So what happened when the cell is dividing? This chromatin fibers, they condense. Suppose this is our chromatin. Okay, this is our chromatin, and when the cell reaches the next stage, this is the interphase stage. Interphase or resting stage. Okay, from the interphase stage the cell reaches the prophase stage. I will be drawing the prophase stage. Okay, in prophase this is the chromatin. Okay, this chromatin condenses like this. It condenses and forms chromosomes. Okay, so this is our cell. Okay, the nucleus also degenerates and this chromatin has condensed and it has formed chromosomes. Okay, these are our chromosomes. Okay, this is the nuclear membrane, and these centrosomes which were helping in cell division, they move towards the opposite poles. Okay, attached by the spindle fibers. These are spindle fibers. Okay, so in prophase we are able to observe the chromosomes. So what are actually chromosomes? Let's write it. Chromosomes. Okay, means color, somes means body. These are colored body. Okay. Now, chromosomes are condensed, coiled chromatin fibers. I hope the definition is clear. They are condensed coiled chromatin fibers. This was our chromatin fiber. Okay. And this chromatin fibers condensed and they formed chromosomes. Okay. Now, let's move to the next topic. Discovery of chromosomes. So, what happened in 1882? A scientist named Walter Fleming, he took cells of salamander's larva. Okay. Salamander's larva. Suppose this is salamander's larva. He took its cells and he observed under the microscope. So what did he see? He saw some colored bodies. He saw colored bodies. Okay, he saw some colored bodies which was named as chromatin. Okay. He took the cells of salamander's larva. This salamander is an amphibia. Okay. What is an amphibia? Amphibia is a class of tetrapoda which have two upper limbs and two lower limbs. So similarly, like amphibia, reptilia, birds, mammals, these are different classes of tetrapoda. So amphibia is also one of the classes of tetrapoda. Last topic, structure of chromosomes. So see how a chromosome looks. The chromatin condenses and forms a structure like this. Okay, these are the two chromatids. Okay, and the point of attachment of chromatid is called centromere. This centromere is a constriction site. Okay, is a constriction site which joins or attaches the two chromatids, which joins the chromatids. Okay, and this centromere also act as a point of attachment for the spindle fibers during cell division. I will be explaining this point also. Act as point of attachment of spindle fiber during cell division. I will be explaining this point. See how. This is our cell. Okay. These are our chromosomes. These are our centrioles. centrioles or centrosomes. Asterase. Okay. So spindle fibers, they attach to the centromere. So this is the spindle fibers. Spindle fibers, with the help of centromere, they are attached to the centrosomes from the opposite poles. Now what happens when the cell is dividing? These centrosomes, with the help of spindle fibers, they contract. Okay, they contract, and these chromatids they separate. Wait, I am drawing again. This is the cell. Okay. So the chromatids have separated like this. Okay, see, you can see the two chromatids, they have separated and they are moving towards the poles. So I hope this function is clear, that they act as point of attachment of spindle fibers during cell division. Okay, now so these chromosomes, they were formed as a result of chromatin fibers only. The chromatin fibers condensed and they form the chromosomes. So that chromatin fiber consists of two parts. First part is the DNA, which is deoxyribonucleic ribonucleic acid. Okay. Then the second part is histone proteins. See, this is our DNA and these are our histone proteins. They occur in octamer. 8 subunit of histones are found. Okay, they both wrap around, means this DNA is negatively charged, it wraps around this histone proteins. Okay, see, this is our histone protein. I am drawing more histone proteins. So our DNA, it wraps around these histone proteins. Okay, and they form the chromatin fibers. Now this DNA, it is 40%. The amount of DNA which is present is 40% and the amount of histone proteins which are present are 60%. Okay, I hope this structure of chromosomes is well understood. Now, before saying thank you, I told you in the starting of the lecture that I will be talking about a book. So the name of that book is Miracle Morning. I will attach the link of its PDF in the description. So just go and check out and just try to read this book in your free time. Believe me, it will definitely help you in achieving your dreams, because most of the people must be under stress because you are giving your boards this year. So I just want you to be calm and move towards your goal slowly and with a very positive mindset. I have also read this book and this book has really helped me in my career. So give it a try and thank you so much. Keep believing in yourself, keep coming and trust yourself. Thank you so much.